Hey, good morning. This is the last session of this series, Introduction to Bioinformatics, and today we have with us Marta Melé. Marta Melé is the group leader of the Transcriptomics and Functional Genomics lab at the Barcelona Supercomputing Center. Marta studied biology in the University of Barcelona, then did a master's in Biomedical Research at the Pompeu Fabra, and then also a PhD in the UPF. After getting the PhD, she did a postdoc first at the Roderick Guigo lab at the CRG, and then a second postdoc at Harvard University. And in 2019, she joined the Life Science Department at the BSc as a group leader. Today, she will present a talk that is entitled The Anatomy of Transcriptome Regulation. So thank you very much and welcome.
Can you hear me?
Yeah.
Ah, okay, cool. Okay, so this is the first time that I'm giving a class to students, so please help me. My fear is that you will disconnect the minute I start talking about difficult things, so please stop me and ask me any questions you want, okay? Okay, so let me see if this works. All right. This is the question that we're trying to address in the lab. How does human diversity emerge? And some students that has come to the lab think that a lot of things are already known, but we don't know why are we different from one another, and what makes us unique, okay? We don't know how this is happening. We have some idea. And why is this important? This is important because everyone is talking about personalized medicine and how we're going to treat the patient rather than the disease, right? This is the focus of personalized medicine. We want to treat the patient and not the disease, but we have no idea what makes each patient unique. So this is what we're trying to address, and that's why this is important. If we want to have personalized medicine and treating each person uniquely, we need to understand what makes each patient unique, okay? All right, so what makes us each one unique? And maybe you can help me here, okay? If we think about the biology of each person, maybe you can tell me what makes, you guys different from me, for example. Can someone guess? Biologically? Yeah? DNA? Okay, your genome, perfect. What else? It's not the genome only, right? I have the same genome if I'm 20, and the same genome if I'm 40, but there is something different here. What else? Yeah? Environment, yeah, very good. Environment. Having healthy habits, whether we exercise or not, whether we smoke or not, whether we drink a lot or not, okay? What else? What also, if I'm a doctor and I want to treat, each of you differently, what else do I need to know? Yeah? Very good. Biological age, that's super important, exactly. And what else? I mean, there's another one that is quite obvious. Well, two more that are quite obvious. And if you look at this picture, you can guess. Sorry? Yeah, genetic ancestry. That's how they call it, but yeah, exactly. Genetic ancestry is very different. And, to treat people from different geographic locations, we need to think about what the genetics ancestry is, yes? And another one that is, that people talk about it a lot in, research and biases and, no. Yeah, sex. I mean, gender is a good one. In our case, we study biological sex, right? Gender is, by choice, how you identify. And biological sex is, whether you have two X chromosomes or one X and Y chromosome, okay? Okay, so this is another one. And, all right, so this is what we study in the lab, okay? We look at all of these factors. And the way we do this is by studying the transcriptome. So we don't study the genome, okay? Because like I said, in age, the genome is the same, but there are things that change. What is it that changes when we age? Or when we smoke, or when we drink? What happens? One of the things that change is the transcriptome. Does anyone know what the transcriptome is, or have you guys any idea what this is? The coding part, the genes, yeah. Okay, close. It's all the RNAs that we have in our organism, okay? All the RNA. So yeah, the genes that are transcribed. The part that is transcribed into RNA is the transcriptome, okay? And why do we care about the transcriptome? Well, the transcriptome is the first cellular phenotype, okay? It's the first thing that happens when a gene is activated, okay? When a gene is activated, this is the central dogma of molecular biology. We have our genome, sorry, whoops. And then it's transcribed into RNA, and some of these RNAs are going to be translated into proteins, or some are going to stay as RNA and have functions as RNA. But this is the first thing that happens. Do we have a pointer? No. No, that's fine. This is the first thing that happens. So we looked at the RNA, and again, why is this important? Okay, all these cells could be from one person, have the same genome, okay? But each one of them is going to become a different cell. What is happening there? Why, if they have the same genome, become different cells? Because the RNAs, or the genes that are activated, expressed, are different between all of these cells, okay? So we really care about RNA because it's going to make a cell going into fat cells, skin cells, nerve cells, or cancer cells, for example, okay? All right. And the idea that I mentioned before, this girl here has the same DNA or the same genome all over her life. But look, I mean, she's very different as time goes by. And this is because the genes that are activated and the transcriptome is going to change through time, okay? Okay, so I hope I've convinced you that it's very interesting to know about the transcriptome and can tell us why are we different from one another. All right, so this is the work that I'm going to be talking to you about that we published this year, where we found, I mean, you know, journalists made it like very flashy, but they were saying that we identified the genes that made unique each person. And this is what they were talking about. We found the genes that were activated in different people with different traits, different sexes, different genetic ancestries, different ages, different BMIs, and different disease conditions, right? When we're sick, the genes that we have activated and the transcriptome also changes, right? Okay. So the study that I'm going to be talking about is going to be looking at genetic ancestry, sex, age, body mass index, which is a proxy for weight, and clinical traits. It focuses on looking at these represent RNAs, different types of RNAs, so the transcriptome, and in a variety of tissues, okay? How can we study the transcriptome? I mean, I don't know if you've done, have any idea about, sequencing technologies and so on and so forth. Have you done any of these? Yeah? Good. So in the beginning, I mean, the transcriptome was studied using microarrays, which really was trying to target specific genes and in a preset, and it was not great. But then there was this next generation sequencing technologies that allowed to sequence all the RNAs in an organism, and then we discovered that we were looking at only a small portion of the transcriptome and that it was way more complex than we anticipated, all right? So RNA sequencing here in 2006, and the word RNA-seq, okay, is started in 2006, and it was, exponentially the number of publications that happens when RNA-seq is shown in black appeared, started to increase exponentially. This is an old plot, but it kept increasing, and it really revolutionized the field of transcriptomics. Okay. So many researchers started to uncover the transcriptome because we can now look at what genes are active in any point in time, in the life of a person, in any tissue, when you're sick, when all of these things, we can now understand what genes were active at each point. Okay, and now we not only have RNA sequencing, we also have single-cell RNA sequencing, which we also do in the lab, and this means that we not only look at all the RNAs that are in the liver or in the heart, we can look at all the RNAs that are expressed in a single cell. So in one liver cell, and ask the question, is this different from the next liver cell or from a neuron or you name it, any cell type that we want. And we also study this in the lab. I won't be talking about single cell RNA sequencing today, but we have several projects on it right now. Okay, and the advantage of single cell, of course, is that you are not only looking at a tissue, at the entire tissue, but you're looking at specific cells every time. Okay. Okay, and what do we do computationally, right? You're going to all become bioinformaticians, computational biologists, so how do we analyze this data? We have all these RNA-seq experiments that experimental biologists do, and then we get a bunch of RNA reads, okay, sequences that we have to analyze. And in reality, this is how we do it, okay? So basically, when we do an RNA-seq experiment, what we do is we take the sequencing reads, okay, the pieces of RNA, and we map them into a genome, and then we extract the expression levels of each gene. So in the end, we'll have a matrix of genes and expression levels. And when I mean expression levels, I mean the amount of RNA that was in the cell, all right? So you need to imagine this as a table of genes in rows and expression levels in columns of the 50,000 genes that we have, all right? And then, when we have these matrices, we're going to have a matrix like this per person, all right? So if we're looking at a cohort of 100 individuals, maybe we want to understand the differences between males and females, or between individuals that are old and young, okay? So what we do is we divide the groups, and we look at, for every gene, whether the expression level of each gene is different between the groups. So this is what we're trying to represent. In this case, is for male and female. In the top, we can see every dot is a sample and a gene, okay? And in the top row, we can see that the gene expression values, okay, here, gene expression values of this gene in males are quite low, and this is the mean, whereas the genes the gene expression of females, which are these dots here, are quite high, okay? So there is a distance between the global mean and the group mean, okay? And in here, there are no differences. And this is what's called differential gene expression analysis. I mean, it's quite simple if you think about it. I mean, it gets complicated when you do it, and there are many things to think about, but this is the basic idea. So if we have two groups, and we have gene expression for many individuals for each of the groups, we can assess whether the genes are significantly differentially expressed, and we put a p-value, and we then know that this is statistically significant, okay? And a lot of the work that we do in the lab is based on this type of analysis, comparing groups and asking, interesting questions that get at understanding what makes each of us different from one another, all right? Okay. Okay, so the first thing that when I started studying the transcriptome, we had this one question that we addressed. This was the first study on transcriptomics that I did ever. And we had a simple question, does the human transcriptome change more between tissues or between individuals? If we take RNAs, the group of RNAs of a liver of one individual and a liver of another individual and a heart from one individual and a heart from another individual, what do you think is going to be more different? Heart to heart from different individuals or heart to liver from the same individual? Okay? It looks like a simple question and something that maybe we should already know, but we didn't know. And what we did to address this question was to use the Genotype Tissue Expression Project, GTEx for short. I'm going to be talking about GTEx. The only thing that needs to come up in your head when I say GTEx is data or transcriptome data for many, many individuals and many, many tissues, okay? Back in 2015, we had 175 individuals and 43 tissues, okay? And we looked at the transcriptome data, and we asked the question, okay, for every gene, can you tell me if the gene changes more across tissues or across individuals, okay? And maybe the answer is across tissues, maybe it's across individuals, or maybe for every gene is different, all right? Okay, so here comes the answer. Every dot is a gene, okay? And this is every dot is a gene. And what we can see is that the proportion of the variation that is explained by tissue is much larger, right, than for individual. If we do the mean of all these points, I mean, it falls right here, right? 0.5. For individual, it's very low, okay? If you think about it, I can take a heart of one person and put it to another person, but I cannot take my own heart and put it in my liver, okay? So this was obvious. We expected something like this. But what's interesting about this analysis, and every doctor would have told us that gene expression was going to change more across tissues, okay? This is obvious. But what's interesting is that there are some genes where this is not the case, and some genes change a lot between individuals. And these genes are important because they can tell us, again, what makes each individual unique, okay? In this case, Okay, so yeah, 47% variation by tissue, 4% variation by individual. In this case, the genes that were at the top, and again, this was kind of expected for some of them, but not all, were related to the Y chromosome. Okay, so these are genes that are only active in males and not in females, so of course they vary a lot between individuals. And there were also genes from the immune system, and the immune system is also very different between people that have genes differentially activated. Okay. In 2019, we could start. Okay, I'm going to stop here. So, from these individual traits that I've mentioned before, now we're only going to focus on inter-individual variation. And I would like to ask you, if we had, a lot of individuals and a lot of tissues, again, now we're only focusing on no differences between tissues anymore, but only individual variation. We have sex, biological sex, age, ancestry, and BMI. And we have all of these tissues. Can you tell me which individual trait, if it's sex, age, ancestry, or BMI, that is going to explain more of the variation between individuals? Do you think there's going to be a trait that is going to be especially different in the transcriptome between individuals? I leave it there. This is the data that we had. So we had genetic ancestry from African Americans and European Americans, males and females, age going from 20 to 70, and BMI from 20 to 35, okay? And the first question that we asked is, which tissue do you guys think, I'm going to put the tissues, okay, from all of these tissues, which tissue do you guys think that is going to be more different between males and females, in general? One of these tissues is going to be like extremely different between males and females. Which one? Reproductive organs are tricky, right? Because females have them and males don't. So it's the extreme case, presence, absence. So that is the most extreme. You're right, but here we have to have the organs in both to compare.
Probably I think it's the brain. Like heard something about the brain.
Okay. Any other thoughts? You were right about reproduction. Which one of the organs that is involved in reproduction we both have? Okay, I leave it there. Okay, and between in genetic ancestry, now which tissue you think is going to be more different between African American individuals and European American individuals? Sorry? Yeah, don't be shy. Yeah, I think I heard it. Skin, yes. Well done. The skin. I'll show you the results later. I'm just asking the questions right now. And for age, what do you think? Which tissue is going to be more different as we age? Brain is a good one, but it's not the one that changes the most. But it's a good one. Heart, very close. It's not the heart, it's the arteries. Okay? Yeah, but good point. All right. So here are the results. The tissue names are abbreviated because that's how we call them. I can tell you all of them. But basically, here are the number of differentially expressed genes. Now you know what differentially expressed genes, okay? This means genes that are different between the groups. Here we have the ones for genetic ancestry, and this is the skin, okay? So indeed, the tissue that is more different between African American and European American individuals is the skin. For sex is the breast, and for age, where are the arteries? Exactly, artery aorta and artery tibial, okay? And this is important. Okay, so skip this, yeah. I mean, we were surprised by this finding somehow, but this guy here, who was a medical doctor from the 17th century, already had this saying, a man is as old as his arteries, and he was right, okay? So the first thing that is, we need to watch out when we age is our arteries. And for me, the most interesting thing is that, okay, many of these things are very obvious, that the skin is going to be different between African and European American individuals, that's obvious. and the breast is different from males and females is obvious. But what is not obvious is that we can now know which are the genes that are explaining these differences, okay? So we find the obvious patterns, and then we can dig in and find the genes that are driving these effects, okay? So I just want to give you one example of one particular gene so that you know what I mean by this, okay? So here in this table, I'm showing, again, all of the tissues on the x-axis, and the dots that are becoming, very small in the y-axis are, again, genes, okay? And I color-coded by the individual traits that are most important in these genes, and I want you guys to focus on this particular gene. What it means, this gene being at the top, is that it's very different between African American and European American individuals. Can this gene tell us something about differences between these two groups? And this gene, turns out it's the malaria receptor, okay? You know what the malaria is? It's a parasite. It kills a lot of people, especially in sub-Saharan African regions, okay? Where African Americans were originally from, right? And African American individuals are less susceptible to malaria infection than European individuals. And why? Well, because they have higher expression of this gene, okay? So this gene is telling us why African American individuals are less susceptible to being infected by malaria than European Americans. Sorry, they have less expression of less expression of the receptor, so the malaria parasite cannot enter, okay? So again, why is this, so this is important, right? We can go from the general patterns. If you told me I'm interested in differences between males and females in the breast, just you go to the paper, find the genes, and you'll see what genes are really explaining these differences, okay? Or any other individual trait that you're interested on, or any other phenotype, okay? So now I'm going to let you guys choose between 3 topics. I can, if you have time, I'm going to talk about all of them, or you can choose. So I'm going to explain to you a little bit about all of the 3 of them, and you can vote, okay, which one you are more interested in. They're all related to what we've been talking about. So I can talk about alternative splicing. Do you guys know what alternative splicing is, more or less? No? A bit, okay. So basically, when we look at the transcriptome, we can talk about how genes are expressed, but also for the same gene, the RNA that is transcribed can be different and give rise to different proteins. So whereas gene expression talks about the level of expression, alternative splicing talks about the colors or the flavor of each gene, okay? This is very interesting as well to look at, because when we did the analysis, the same analysis, age, sex, body mass, in everything, it turns out that alternative splicing behaves in a completely different way than gene expression, okay? So, all right, I can also talk about clinical traits, because in this GTEx cohort that I talked about, in principle is a healthy cohort, but we're not always, I don't think anyone is completely healthy, okay? We always have some things at some time, and of course this cohort, some individuals had some conditions, and we also looked at that. Most of the work is focused on diabetes because it's super prevalent. I'm sure most of you, does anyone know someone that has diabetes? Yeah, right? Many of you might know because this is super prevalent, okay? All right, so a lot of work was, in this case, I'm going to be talking about how diabetes affects the expression of your genes. And finally, the effects of tobacco smoking in your tissues, like you said, environment, habits, right? What we do influence how genes are expressed, all right? So, okay. So who wants me, maybe I'll talk about all of them if I have time, eh? No, I won't have, no? So, who wants me to talk about alternative splicing? 2 people. 3 people? 2 people, all right. Who wants me to talk about clinical traits and diabetes? 6, 7? 7 people, all right. Who wants me to talk about tobacco smoking? Yeah, I would guess so. I know. Every time that you do this, it's the same answer. Okay. All right, so I'll skip splicing. Okay. So this is work in the same cohort of individuals. So we have 46 tissues. It's the same tissues that I showed you before, okay? And we have information about gene expression, alternative splicing for all of the tissues, also DNA methylation. Do you know what DNA methylation is? Okay, so this DNA methylation are modifications in the genome, okay, on the DNA that may have an impact on gene expression, okay? And DNA methylation, the environment affects DNA methylation a lot, and smoking, we were guessing, was going to have an influence on DNA methylation as well, okay? So that's why we looked at DNA methylation. And we also used tissue histology. So for all these tissues, and all these donors, we had images. And now what's happening with image analysis that is revolutionizing, you know, pretty much everything? Who can tell me what technology is now revolutionizing, you know, how we look at images and how we identify objects in the image? AI, yes, exactly, AI. So now we can, I mean, in our case, we use artificial intelligence to analyze the images of all of these tissues and understand the effect of smoking across the tissues, okay? All right, so again, how does cigarette smoking impact the transcriptome? Who can tell me the tissue now that is going to be most affected? You all should know that. Yeah, the lung, all right. So that's expected. If we did the analysis and we didn't get the lung, we would know that it was wrong. I mean, this is not the case. But the surprising finding, right? I mean, science works like this. You have your expectations, you find what you expect to find, and then you find something that is like, what is happening here? And this happened here. So the second tissue that was most affected, can you guys guess? I mean, I didn't know, like, at all. I could not have guessed. I'm going to give, yeah, you have all the tissues here. Just say them randomly, because even if I encourage you, you won't get it, I don't think. Yeah? No. Sorry? Yeah, we were hoping arteries would be the one. It isn't. We have salivary glands, and we didn't see it. I mean, mind-blowing, because... Okay. Any other guess? No. Brain? No. This is a good point, because brain, you know, there is this addiction, and, you know, it could be brain, but it's not brain. Skin, not skin. Okay. Sorry?
The esophagus or the
The esophagus mucosa. So the esophagus mucosa is the third most affected. So well done. The third most affected. Yeah, good job. All right. So I'm going to show you the results. So the tissue that was second most affected was the thyroid, okay? The thyroid controls many things on the metabolism, controls growth, and we were very surprised by this finding, and of course we tried to dig in a little bit more and understanding why the thyroid is affected in smoking. And of course there is literature talking about it, but for us it was the first time that we put into perspective the comparison between the different tissues. So lung is number 1, thyroid number 2, and esophagus mucosa number 3, all right? But what else is interesting about this plot? I mean, what is it telling us? So here we have the number of samples, okay? Number of samples. Never smokers, we have also ex-smokers and smokers, and the comparison is between smokers and never smokers, and on the top we have the number of differentially expressed genes in every tissue. What's interesting about this plot? For me, what was cool is that it's not only the lung that is affected, right? I mean, pretty much all of the tissues are affected. Even tissues where the smoke, it doesn't get there, or it's not directly, you know, tissues like, you know, the heart. I mean, the heart doesn't get in direct contact on the smoke. It means that the toxics of the smoke get into the circulatory system and affect the tissue, okay? So the main finding, I mean, of what this plot is telling us, apart from that the thyroid, you know, something is going on in the thyroid, is that, you know, what I said in the title, cigarette smoking has a systemic effect, all right? If we look at the genes, are there genes that are differentially expressed between smokers and non-smokers systematically across most of the tissues? And here we have these 3 genes that are differentially expressed in 30 tissues. So if you smoke, these genes are going to be changing a lot, no matter the tissue that you look at, okay? These genes are involved in the metabolism of toxics of smoke. So this is one telling you, like, the toxins of smoke reach all of the tissues in the body. All right. And then the finding of the thyroid, that to us was very striking. What is going on in the thyroid? And how could we look at, what's going on in the thyroid? What could we do to understand what is going on in the thyroid? We've looked at the gene expression. What else could we look at that I told you that I had information on? Gene expression, what else? Splicing, good point. We looked at splicing, nothing interesting. What else? For this cohort, we said we have gene expression data, methylation, and what else did we have? Images, no? We had images of the thyroid of these donors, and we have artificial intelligence to tell us what is going on. So this is what we did, okay? I mean, it is a possibility, right, that you have a finding that is interesting in science, and it could be that there is some other factor that you don't know what is going on, that could be, making the results being wrong, okay? Maybe there was something in the thyroid, or maybe there was what's called a batch effect, and maybe what happened is that the individuals that had, that the smokers were processed in a different way. So we wanted to know if this was really true, and if it was true, in principle, a classifier or a model that looks at the images should be able to classify if the signal is true between smokers and non-smokers, okay? If it was something else that we didn't understand because someone processed the samples wrong, the biological signal would not be in the images. Would be in the transcriptome analysis, but not in the images, okay? So what we do is to use deep learning and the typical convolutional neural network architecture. I mean, I'm not going to go in details to this, but basically what we do is we take huge artificial intelligence models, and we use them, and we use the images to train them the last layer of these huge network models, okay? And we ask the model, can you classify between smokers and non-smokers? Okay, and the answer is, this is a ROC curve. The answer is for thyroid, okay? The ROC curve needs to be read like this, so that the higher the area under the curve, the better the results. So these models were able to classify between smokers and non-smokers, just looking at the images of the thyroid, better even than for lung, and better even than esophagus mucosa. So something in the images of the thyroid of smokers was different, okay? And then, of course, we can take the extremes. This is for the lung, and what we can see in the lung visually is that the smokers has macrophages in the lung, and this is how the thyroid of the smokers look like, okay? If we take the probability of being classified as a smoker by these models, and you look at the images, what you see is that the follicles of the thyroid are much larger, okay? Okay. And, okay, so I've told you that smoking has huge impact on all of the tissues. How do you think the ex-smokers look like? Can anyone guess how the tissues of an ex-smoker look like? Do you think they're going to be, similar to a non-smoker? Or they're going to be the same as a smoker? Or somewhere in between? In between, huh? Yeah. So for gene expression, that's exactly what we see. And in fact, when we look at gene expression, if we ask a classifier to classify ex-smokers into a smoker or never smokers, most of them are classified as never smokers. So gene expression goes back to normal very fast. But I told you that we also had information about methylation. And for methylation is different. So the methylation, it takes much longer to go back to baseline levels, okay? So the modifications on your genome that change the methylation status by smoking take longer and more time to go back to baseline levels, all right? Okay, and then there is a lot of literature about this, whether smoking makes you age faster There are even, I mean, I found, one paper that was looking at twins, and they were taking pictures of the twins. One was a smoker, one was not a smoker, and the classifier, the classifier was considering, the twins. The classifier was considering the twins older, the smoking twin older than the non-smoker. Okay, so there is a lot of literature out there saying that smoking ages you, but in gene expression we didn't know. Okay, so we asked the question: is smoking making your tissues like they are older? And I don't know. Tell me what is your thoughts? Do you think or that this happens or not, or can you guess? Yeah? Okay, so this happens in some tissues, okay? So we took the genes that are differentially expressed between old individuals and young individuals, and the genes that are differentially expressed between smokers and non-smokers. And what we saw for these tissues over here is that the genes behave in the same way. So for example, you take a gene, if you are older, it's going to be upregulated, and if you're a smoker, it's going to be more upregulated. Okay? And for other genes, same thing. If the gene is downregulated in smokers, it's also going to be downregulated in old individuals. Okay? So what this plot is trying to tell us is that the direction is always, I mean, in red, okay, is almost always in the same direction. Okay? So yes, the answer is that when we look at the transcriptome, we see that smoking ages your tissues, basically. And the same happens with methylation. We see this in two tissues, in the lung and in colon. Oh, yeah, this is from. All right, so how am I doing on time? Done. 5-10 minutes. So I'm going to read the conclusions. Okay. All right, so ancestry, sex, age, and body mass index have a significant impact across tissues. In the transcriptome, and we've seen that their contribution is tissue-specific. For sex, it's breast. For genetic ancestry, it's the skin. And for age, it's the arteries, but they all affect all of the tissues, okay? Even if you have the same genome, this is happening. Okay, and this is, yeah. And then the smoking bar. Cigarette smoking has a systemic effect on the transcriptome, and the strongest effects are in lung and thyroid. All right, and I want to thank my lab because they are the best. I don't do any, programming anymore, so they all do the work. Yes, and I can take questions.
Thank you very much for this fascinating talk. So now time for questions. We start, as always, with people in the priority list, and then we open the general turn.
So these are compulsory questions? Yes. Yeah, okay. Good.
So, after all, can we look for any pattern that, besides it is not so accurate, we can use it when it comes to personalized medicine for a huge amount of people?
Sorry, I couldn't hear the first part.
Like, if we can look for any pattern that, besides it is not so accurate, we can use when it comes to personalized medicine for a huge amount of people.
So you mean if we can use this data to look at any pattern?
For some pattern to make personalized medicine more affordable, like for race, for...
I mean, I think it needs to be, this personalized medicine, it considers like a lot of different things. So I think we should go into the case-by-case basis, and depending, for example, on the drugs, maybe genetic ancestry matters more, or aging matters more. If you think about it, like if you have to take a drug for your arteries, then what demographic trait would matter? Age, right? Because they change a lot. So it's going to be depending on the tissue, depending on the drug, and depending on the disease. What I was trying to make the point is that we need to understand, in health what happens to then be able to treat the patients, right? But this is, personalized medicine is like, huge thing, and I think we need to kind of answer this question going disease by disease.
Okay. Well, my question is that how can functional genomics and transcriptomics be used to identify new therapeutic targets, like for cigarette smoking related diseases?
Yeah, so I mean, if you find the genes, right, that are changing with the disease, like I don't talk about diabetes, but in diabetes we find the genes that change in the nerve, and the nerve is, in diabetic patients, the nerve changes a lot, and there is a condition that is very serious that is called diabetic neuropathy. And we identified the genes that were different in diabetic patients that had diabetic neuropathy. Some genes were known and some were novel. If you identify the genes that are changing in disease individuals, these are therapeutic targets, okay? But then, I mean, as researchers, at least in my lab, we find the genes and then we give it to, this knowledge to whoever, pharma companies and medical doctors that can really, bring this to the clinic. So this basic research, you understand the mechanism and what happens, and then identify the genes. And then these lists of genes are super important because then we know what needs to be targeted for each disease.
So what do you think that are the future challenges that are faced in the study of transcriptome regulation?
That's a good question. For me, right now, what I think we are missing is that we are always looking at the same kind of people. We're always looking at, individuals from 20 to 70, individuals from European ancestry, okay? I mean, we had some African Americans, but what about, South Asian or East Asian or, Native American population? So we need to kind of understand the transcriptome of, or what about kids, right? I mean, what about postnatal? And so I think that the challenge now is to get at the full worldwide population, because you want personalized medicine to be for everybody. We cannot just focus on European and males and no, I mean, this needs to be for everyone. So I think for me, it's not the next challenge in transcriptomics, it's the next challenge in science in general. I mean, biomedical science in general, this needs to happen.
Yeah.
Based on your study, which is the tobacco smoking impact that is more that surprises you the most and people don't know about it?
So the one that surprised us the most was the thyroid. And also, I didn't talk about this, but the pancreas also shows quite a lot of signal. So those were maybe the more surprising findings, and the aging thing, because, there's literature, but it wasn't very solid, this aging idea of smoking. I thought it was more like advertising to have people stop smoking somehow, but we see it very clearly. So yeah, that would be maybe the most striking pattern, yeah.
Following the subject, it is known if the electronic cigarettes cause the same impact in transcriptome that smoking?
Yeah, we have no idea. Like no idea,
Okay.
no. It's a good question. I've had several scientists ask me, when are you going to do the cigarette smoking thing? I mean, it's very challenging to have a cohort like this. These are dead donors, eh? So, there has to be, I mean, it's not that easy to get information about, electronic cigarettes, but it's a good question. Maybe there will be soon studies in whole blood, and then from whole blood, maybe we can extrapolate what the consequences are and compare with whole blood with the smokers. Yeah, but it would be very interesting to look at that, yeah.
Thank you very much again.